Hello everyone and welcome to Instant Biology by Dr. Neelab. Today we are going to talk about a new topic in the lecture series of analytical techniques. So as you already know that we have multiple videos in the lecture series of analytical techniques. This would be an addition to that. A very important topic that you all should be aware of. This is asked in different life science examinations like GATE and CSIR net. So today we would be talking about DNA microarray. So basically what this technique is basically helping us to analyze the gene expression of multiple genes at a single time. Okay, so this was just a brief overview of what we are going to study in this lecture. So let us start with each point in detail. So what is DNA microarray technique? What are the other names of DNA microarray technique? This can be asked in by any name. So gene chip, DNA chip, bioarray and gene array. So these are the names. Let's see again: gene chip, DNA chip, bioarray or gene array. These are the different names of DNA microarray technique. Why are these names, or why such names have been given? You need to understand this, that there is a chip. You might have seen chip in a circuitry, right? If you have seen any kit, if you have seen any machine diagnostic tool, there are chips that are present on the tool. So over here also there is one chip that is the key to all functioning or key to all mechanism of this technique. I would be telling more about that chip. So that particular chip, suppose let us say that this is a chip. Imagine that this is a chip, and on this chip there are multiple genes that we have attached. Okay, multiple DNA fragments that we have attached. So that because of that, this is called as gene chip. DNA, because the gene is made up of DNA chip. Or it is also called as bioarray, right? Because this is microarray, in the same sequence it is called as bioarray or gene array. Okay, array means basically sequence, right? So there are multiple sequence of the genes that are attached on a chip. That is why we are calling it by these names. Okay, so this was the first point. Next point is, what are we looking at over here? So there is a solid support. It can be made up of glass or silicon. So what is the solid support that we are talking about? This can be the solid support. Okay, so this is a chip. This is a rectangular chip. Basically it can be of any shape, but let us take that it is a rectangular chip like this. So this is the shape of the chip. It can be made up of glass or it can be made up of silicon, both ways. I mean, it can be made up of glass also, it can be made up of silicon also. So this chip, or it can also be made up of a nylon membrane, but in general glass and silicon chips are used. Okay, what does this chip have? It basically has thousands of genes from a given tissue. Now let us say that we are talking about liver. Now you would be knowing that liver in particular would be expressing number of genes, isn't it? For its own function, this liver would be expressing number of genes. For example, genes would be expressed for the formation of bile. Example, I'm just quoting an example. So these genes are specific and they express in liver only. They do not express in your eye cells. That means the formation of bile does not take place in your eyes, isn't it? So these cells or these genes are very specific to a given tissue. Suppose I am working on liver. I just gave you an example. So I would be dealing with the genes that I have in my, or that the genes that are expressing in the liver cells, isn't it? So what happens is that there are orderly arrangement of thousands of genes. If I am working on liver cell, then whatever genes are expressed inside liver, you should understand this, that the DNA of all the cells, be it liver cell, suppose this is a liver cell, suppose this is an eye cell, suppose these are cells from your stomach, or these are cells from your kidney, or these are cells from your neurons. All of these cells, the neuron does not look like this, it looks like this. Okay, I'm not drawing it to scale, just understand just that these are the dendrites and this is the axon. So all of these cells have the same type of DNA. The DNA in all of the cells is the same inside every given individual. So what is the difference? The difference lies in the gene expression. That means that each cell, depending upon which location it is present, depending upon whether it is present in brain, whether it is present in liver, whether it is present in eyes, it expresses different types of genes so that it can perform the given functions of that organ. Okay, so for example, I gave you the liver, right? So liver forms bile, so the cells of the liver are expressing that kind of genes that would form the bile. Okay, suppose I'm talking about let us say the brain cells or neurons, for example. So neurons would be expressing different types of genes so that they can help in these cells can help in transduction of the signal. Okay, because every cell is doing different kind of functions, therefore different types of genes are expressed. However, the DNA of all the cells is the same, just the gene expression varies. This is the key point, take home point from this lecture. Okay, so now depending upon what we are working on, suppose I'm working on liver, I just told you. So what I'll do is I will attach thousands of genes. Suppose liver has thousands of genes that are expressed in it. So I will attach all those genes on this very small chip. You might be looking at this chip and saying that how many genes can get attached on one chip, but we do it automatically, so there are thousands of genes that can be attached. Okay, so orderly arrangement of thousands of genes on this DNA chip. So the DNA that is responsible, or that is that particular gene that is attached on this chip, this is up till here. Okay, thousands of genes. What is the purpose? Why are we doing so? We are doing this in order to understand, in order to learn the expression profiling of those thousands of genes simultaneously. That means, suppose I want to check for a disease inside a liver. Okay, suppose I want to check for a disease inside a liver. I have the gene sequences, I have the information or data of a normal individual, and I have the information or data of a diseased individual. What I can do is I can compare the gene analysis, I can compare the chips of those two individuals, the individual who is normal, the individual who is diseased. I can compare both of them and I can tell that if any genes are overexpressed or underexpressed. Overexpressed means they are expressed more, underexpressed means that they are expressed less in comparison to a normal individual. Now you would understand this, that if the genes are expressed more or less, they would result in some kind of a disease. So this information can help us for analyzing the expression profile of thousands of genes at one instance only. You do not need to do the experimentation again and again. You just need to do it on one chip, and all the thousands of genes you can analyze at one time only. You do not need to re-perform the experiment. This is the best benefit, or this is the best advantage of this technique. The next one is can also be used for protein DNA interactions. So over here I just told you that we can analyze the gene expression. You can know about the which genes is expressing more, which gene is expressing less, but it can also be used for protein DNA interaction. Now you would already be knowing that suppose this is one DNA, isn't it? Now on this DNA there are protein binding sites, isn't it? Protein binding sites are present. For example, you would be knowing that transcription factors, they bind to a particular region inside the DNA and they can enhance or repress the transcription. So that is DNA protein interaction only. You can also know about the DNA protein interaction with this technique. You can judge what is the transcription factor, what is the transcription factor binding site with this method, or you can also tell that whether the transcription factor is binding to the DNA or it is not binding to the DNA, whether it is binding at the correct transcription factor binding site or not. You can also judge with this method. Next, alternatively spliced variants. So you would be knowing that there are two types of splicing, right? So alternative splicing and constitutive splicing, these are the two types of splicing. In alternative splicing, we know that different combinations of the spliced fragments are joined together. So you can understand that whether you can study the alternative splicing also through this kind of method. However, I am not going to explain alternative splicing over here. This would be a very long lecture, however. So I would stick on to this microarray only. Epigenetic status of the genome can also be studied through this technique. What is epigenetics? So whenever the environmental factors, they affect the DNA, what is the gene, what are the genes that are being expressed, that is called as epigenetics. Okay, so that can also be studied through this microarray method. Next, DNA copy number change. You know that DNA copy number means the amount of the DNA, the copies of the DNA that are present. So you can judge if there is a change in the DNA copy number through this technique. Sequence polymorphism. So what is morph? Morph means form. Polymorphing means number of forms of a given sequence. So there are small changes in sequence. Suppose this is sequence number 1, sequence number 2, sequence number 3, sequence number 4. So these 4 sequences are varying amongst each other in a very small way. So you can also study those sequence polymorphism through this method. Okay, so these are the, I mean, in broad terms, these are the purposes, or these are the main areas where this microarray method is used. I would also tell you about the applications. I have written over here some of the applications, so we'll talk about those also. But for now, you can understand this is the main thing. What are the different types of DNA microarray? Understand this, that DNA microarray can be divided into 2 major portions. So what are the 2 major portions? Depending upon the solid support that I was talking about over here. So what is the solid support that you are using? This is the main thing. If you are using a glass DNA microarray, that means you are using a regular glass slide. Okay, that is called as low density DNA microarray, and this uses glass slide. Why are we calling it low density? Because the number of genes is limited over here. Okay, there is less space. I mean, I'm not saying that there is less space, but the number of genes that you can imprint onto the glass slide over here is less. That is, lesser density is present. That means the area and the number of the genes that you are attaching in that given area has a lesser density. Number of genes over here can be less. So lesser density, and the material used over here is glass slide. Okay, so there are 2 methods in which you can do that. You can use a pen tip method, and you can also use a inkjet method, that is spraying. Okay, inkjet method and pen tip method can be used to attach your DNA fragments onto the glass slide in this method. However, you do not need to go into the depth that how these glass slides are made. You just need to understand the mechanism and what can be done, what can be achieved with this kind of a technique. Okay, so now let us move forward and talk about the high density oligo microarray. High density oligosaccharide microarray. Oligonucleotide, sorry, I'm sorry. High density oligonucleotide microarray. So high density means over here, as you would understand that over here what will happen, the number of genes in a given area would be more. Over here it was less, over here it would be more, so it would be higher density. What is the method that is used over here? Gene chips are used over here, and high spot densities I've already written. Approximate density is 10 to the power 6 spots or 10 to the power 6 genes in 1 to 2 centimeter square area. So this is the density. And there is a very common there is a company called as Affymetrix. It manufactures an item or a product called as gene chip. So this is in general used for the high spot densities oligonucleotide microarray. Okay, so I believe you would have understood this. Now let us talk about the working. How does this work, or how does this microarray function? So the main or the basic principle over here is the Watson and Crick base pairing. The principle that on which this DNA microarray functions is Watson and Crick base pairing between the target and the probe. Okay, so you already know that DNAs can complement with each other, isn't it? Because of the hydrogen bonding that can be present between the two DNA fragments, they can interact or they can be attached with each other, they can hybridize with each other. This is the principle of behind this particular technique. So what is the target and what is the probe, we will be talking about in just a minute. Okay, so this has been evolved from the southern blotting. So you would be knowing what is southern blotting. In case of southern blotting, what is analyzed? DNA is analyzed. Over here also in general you are working on DNA only, you are working on the genes only, you are studying the gene analysis or gene expression. That is why you are calling it is evolved from southern blotting. Northern blotting is for RNA and western blotting is for protein. Only southern blotting is for DNA. That is why we are using the term southern blotting over here, or it has evolved from the southern blotting. What are the steps that are being used over here? Sample preparation and labeling, hybridization, washing and image acquisition. So these are the steps that we would be talking about the working with the help of this diagrammatic representation, and you would understand each and everything. So let us first of all talk about that how this is done. So suppose I have sample number 1 and I have sample number 2. Okay, so suppose this is a healthy individual, sample number 1 is a healthy individual and sample number 2 is a non-healthy or a ill person who is not well. So suppose there is some kind of a issue in the liver, some kind of a disease in the liver. Okay, then that might be because of the overexpression or underexpression of the genes of the liver, that we have to judge. Okay, so now what we are doing is we are taking out some of the RNA from the liver of individual 1 also and individual 2 also. You can isolate the RNA from the liver. Okay, so what is this? This is the RNA fragments. These are the RNA fragments that you have obtained from the liver of the normal individual and of the diseased individual. Okay, then what you are doing? What you are doing is you are forming the complementary DNA of this messenger RNA. So you would be knowing that how you can form the complementary DNA from messenger RNA. You can form the complementary DNA by the reverse transcriptase enzyme. What you use is you use reverse transcriptase enzyme. Just let me just rub this. So what you use is, so this is the DNA, this is suppose this is the RNA, and from the RNA if you want to form the DNA, this is the complementary DNA, and this complementary DNA would be made in double stranded. So this would be double stranded DNA. Remember, this RNA formed this DNA, but this RNA formed this DNA. That means this RNA and this DNA strand, let's say this is strand number 1, this is strand number 2. Okay, so this RNA formed strand number 1, so RNA is complementary to this strand number 1 of complementary DNA, and then we from one strand, from strand number 1 we formed strand number 2, because DNA can be of double stranded nature, isn't it? You got this? And now you would be able to understand that this second strand would be exactly the same as that of the RNA, isn't it? This second strand would be exactly the same as that of the RNA. Okay, you would have got this. So what we will do, we would make use of this particular strand. You would make use of this particular strand, and what else you would do? You would label it with a fluorescent dye. What would you do? You would label it with a fluorescent dye. So what dyes I have used? I have used a red colored dye and I have used a blue colored dye. However, you generally do not use a blue colored dye, you use a green colored dye, but I did not have the green colored pen, so I used a blue color pen over here. Okay, so what you do is you have labeled, you have taken this complementary DNA strand, this lower one that was exactly the same as that of RNA, so you have used this strand and you have labeled it with red color fluorescent dye, and this one you have labeled with, let us say, green colored fluorescent dye, or for my case I have used a blue colored fluorescent dye. Then what you do is, so you have the complementary DNA that was exactly same as that of RNA. You have this chip. I told you about the chip. This is the solid support, and I have attached genes over here. So these are the black colored portions are the genes. Suppose all of these genes are present inside the liver. Okay, now what you are doing is you are allowing this and this to be incubated on this gene, on this gene chip, on this chip. You are allowing this and this to incubate on this chip. Now what will happen, because of hybridization, this is DNA, this is also DNA, and this DNA has been obtained from this RNA. You already know that RNA and DNA would be complementary to each other. That means this strand and the strand that is present over here would also be complementary to each other, isn't it? Now after incubation, the next day that you come, suppose 24 hours incubation was there, let us say that. So after incubation, when you come on the next day and scan and take image, you would see that certain regions or certain dots or certain genes are still black in color, certain genes have turned red, certain genes have turned blue, and certain genes are a mixture of red and blue. So if I would have this green pen over here, then green and red I would have combined to make a yellow color. Okay, I believe you would have understood this. But over here, suppose in this case, this black one, you can see this black region. So this black region means that it was not expressed, because it did not take any color. It did not take the red one, it did not take the blue one. That means this gene was not expressed either in the normal individual also, in the ill person also, in the unhealthy person also. This gene did not express. Same thing was happened with the second number gene. But if you look at the third number gene, this is blue in color. This means that this gene started expressing in sample number 2, or the ill patient, but did not express in sample number 1, or the normal individual. Now coming to this last gene over here, you can see that this is being expressed in the normal individual, sample number 1, but not expressed in the unhealthy individual. Okay, so in this one, let us talk about this one. Okay, this gene. So this gene is a combination of, let me just try and draw this in front of you. So I am drawing it red also, and I am mixing some blue also over here. Okay, that means if I'm saying that if I had a green colored pen, then I would have mixed the green and the red one and made a yellow color over here. Okay, but for understanding, I'm saying that this gene is being expressed in sample number 1 also and sample number 2 also. I believe you would have understood this. These two genes are only expressing in sample number 1, these two are only expressing in sample number 2. So with this kind of data, you can easily analyze that which genes are expressing in a normal individual, which genes are expressing in a diseased individual. Now one thing more, I said that you can also analyze, this was just the analysis of which genes are expressed, which genes are not expressed. I also said that we can analyze that which genes are overexpressed, which genes are underexpressed, which genes are not expressed. How can we determine that? Understand this, suppose one gene is very active, it is very active gene. When it is very active, what will happen? There would be a greater concentration of the RNA. RNA would be formed more. RNA would be formed more. There would be a greater concentration of RNA. This would lead to a very high concentration of fluorescence. Why? Because this RNA would lead to the formation of this complementary DNA, and a lot of complementary DNA would be binding to this chip. Because of that, there would be a lot of fluorescence. So whenever a gene is active, higher amount of RNA would be formed, greater amount of fluorescence would be seen. Less active gene, less amount of RNA, the fluorescence that would be obtained would be dimmer. If a gene is inactive, no messenger RNA would be present and no fluorescence would be seen. So this is the concept. Now you can easily understand that which gene is being expressed or not, and you can also analyze that what is the amount of the gene that is being expressed. I hope this is very clear to you. Now coming to application portion. What is the application portion? That first of all, you can easily analyze that, you can easily investigate the cellular state and processes. What do we mean by a cellular state and processes? Now understand that there are different states. Okay, when a cell undergoes different stages. For example, let us say cell division. Okay, cell division has different stages, and in each stage there are different genes that are being expressed. You can understand with this microarray that what is the stage of the cell. Suppose when I talk about sporulation, the spore formation or sporogenesis, okay, what happens is there are different phases of that. So in that you can easily understand that which phase is the spore in with the help of this microarray, because with the help of the gene that are being expressed, you can analyze which region or which phase the spore is in. Diagnosis of a disease, I told you the entire thing, I gave you the example of diagnosis of a disease only. You can understand the difference between the sample 1 and sample 2, the healthy individual and the non-healthy individual. Drug selection, what is this? Basically you can analyze the genes of an individual and understand that which gene or which drug would be more effective in case of this individual by just the analysis of the gene. You might have heard about the tailor-made drugs or personalized drugs. That is the future. Okay, we can also work on that. Specialized diagnosis of the disease is basically the same thing, but I just want you to understand that specialized diagnosis, there are different types of cancers. Okay, there are different types of cancers and there are different stages of the cancer also. Suppose there are different types of breast cancer also. Let us take the example of breast cancer. So what is that type of breast cancer, or what is the type of a given cancer you can diagnose with this method, specialized diagnosis of the disease. Last one, pathogen resistance. You can understand, now you would be knowing that there are different types of bacteria, there are different types of fungi that are becoming resistant to the current drugs. You can understand that which genes are conferring the resistance inside them, and then you can work on probable drugs or probable therapies so that we can make them less resistant, or we can kill those pathogens. Okay, so this was all in all about microarray technology. I believe you would have understood this, and if you have understood this, kindly like the video, subscribe to my channel and press the bell icon so that you can stay connected with me. Join us on Telegram also. There are regular updates on Telegram as well. Thank you so much and have a good day.